Hi my dear friends, in this video we are going to talk about the hairy cell leukemia, and it is very important for your PG entrance examination point of view. So friends, let's begin. So what is hairy cell leukemia? As the leukemia word is a misnomer, as it is not leukemia, it is actually a B cell non-Hodgkin lymphoma which occurs in old age people, and these hairy cell leukemia are associated more in the affects more males as compared to the female. So male is to female ratio is 4 is to 1, and it is characterized by the presence of hairy cells in the peripheral blood, and it is associated with the splenomegaly and the pancytopenia. Now friends, the exact cause of this cancer is unknown, but the role of tumor necrotic factor alpha is postulated, and which is responsible for the proliferation of the hairy cells and are responsible for the suppression of the proliferation of normal bone marrow cells, which causes pancytopenia. And the chromosomal abnormalities, if we talk about the mutation, then it is associated with the trisomy of the fifth number chromosome, and at the fifth chromosome there is a proto-oncogene, and because of that there occurs excessive proliferation. And now if we talk about the clinical features, so it is associated with the massive splenomegaly, and but in the hairy cell leukemia from this age it is related to hepatosplenomegaly, but the hepatosplenomegaly is very rare. Most commonly find clinical feature is the splenomegaly and massive splenomegaly, but rarely there is presence of hepatomegaly. And also lymphadenopathy is distinctly rare in this disorder, and because of the bone marrow failure there occurs pancytopenia, and because of the pancytopenia the lymphocytes level decreases, and because of this lymphocytes level also decreases, and the leukocyte and all the leukocytes level decreases, and these are for fighting against the infection. So if these leukocytes are less, then chances of infection increases. As this leukemia or lymphoma occurs in old age patient, that is why it causes fatigueness in the patient, and as the thrombocytes level is less, and because of this there occurs bruisability in these patients. Bruisability means, if let's suppose this is the individual, and if there occurs either blunt injury or any type of injury in the body, then it causes bleeding under the skin, and because of this bleeding some changes start to occur in the at the site of the trauma or at the site of the blunt injury. So after bleeding there will occur a blue, and then the blue will changes into the black. So at the site of injury, at the site of trauma, first there will occur a bleeding, and that trauma will changes into the blue color first, then later on it will convert into the black. Now friends, if we talk about the investigation, so first there is blood, so there is pancytopenia. So friends, in the blood picture there is pancytopenia, there is presence of atypical lymphoid cells, presence of leukemic cells which are hairy cells, and this is the characteristic feature of the hairy cell leukemia, and these are the characteristic cells, and these characteristic cells are hairy cells and which are leukemic cells. Now these hairs are because of the presence of cytoplasmic projections which looks like hair, and these hair like projections are seen under phase contrast microscope, and under electron microscope there is presence of ribosomal lamellar complexes in the cytoplasm. So for looking here in the cells, phase contrast microscope is needed, and for the ribosomal lamellar complexes in the cytoplasm, electron microscope is needed. And the peripheral blood shows presence of hairy cells with nuclei of different shapes. So in some hair cells, the shape of nuclei will be like this, and in some hairy cells, the shape of nuclei will be like this. So these nuclei are of different shapes in different hairy cells, and these hairy cells are leukemic cells. And these hairs are seen under the phase contrast microscope, and under electron microscope there are ribosomal lamellar. These are the ribosomal lamellar complexes seen under the electron microscope. Now friends, if we talk about the bone marrow aspiration, so in the bone marrow aspiration, what we get? Dry tape, and this is because of reticulin fibrils which are present along with the leukemic cells, and these leukemic cells are hair-like cells, and because of these hairs they form a network. And because of this, if we insert needle in the bone marrow, then we find no cells in the aspiration, and that is called dry tape. If we take bone marrow biopsy, then it reveals the infiltration by the cancer cells called the honeycomb appearance, and the leukemic cells have a nucleus surrounded by the cytoplasmic halo, and that is called fried egg appearance, which is the diagnostic of hairy cell leukemia. So because of these hairs that are attached to each other by the leukemic cells, and they form honeycomb like appearance, and the cytoplasm of the hairy cells or leukemic cells is transparent, or that is halo. That is why, and this is the nucleus, and this is the cytoplasm, and this cytoplasm looks like a halo or glass like. That is why it is called as fried egg appearance. So from the hairy cell leukemia, this H in the hairy will remind you that it is related to honeycomb appearance, and in Hindi hair is called fur. Hair is called fur in Hindi. So from this fur you can derive this word fried egg appearance. So honeycomb appearance and the fried egg appearance is seen in hairy cell leukemia, and this is the diagnostic of the hairy cell leukemia, and this is very important MCQ. Now friends, if we talk about the immunophenotyping, so these are the B cells. So they will have CD19, 20, and also they have 11C, 25 and 103, and this is positive only in hairy cells. So tumor cells have the presence of B cell markers like surface immunoglobulins. CD19, CD20, CD11C is not present in the B cell, but it is present in the hairy cells. CD11C, CD25 and CD103, and these all three are present only in the hairy cells. That is why presence of CD11C, CD25 and CD103 are present only in those cells which are present in the hairy cell leukemia. And these hairy cells are also TRAP positive, and TRAP means tartrate resistant acid phosphatase enzyme is present in these cells. And TRAP positive was also present in the marginal zone lymphoma. So TRAP is also positive in some of the cases of splenic marginal zone lymphoma. Hairy cell leukemia appears benign even though it is malignant because it is having less mitotic rate, resulting in reduced nucleus to cytoplasmic ratio, while in the normally malignant cells the nucleus to cytoplasmic ratio is very high. That is why it is a benign tumor. And this hairy cell leukemia, between the cells of the hairy cell leukemia there is a space present between the tumor cells, while in the malignant cells the cells are overcrowded. So friends, this is all about the hairy cell leukemia. I hope this video will be very helpful for you, and if you really gained something from this video, then please share this video as much as possible to your needy friends. And in the last, thanks for watching this video, and I wish you all the best for your bright future.